So welcome and thank you for coming to this webinar series from the Eye and Ear Foundation called Sight and Sound Bites. We do this every other week. We highlight research at the University of Pittsburgh School of Medicine. I'm Lawton Snyder, CEO of the Eye and Ear Foundation. The Eye and Ear Foundation supports research to advance care for vision, hearing, balance, voice, and cancers of the head and neck at the two world-renowned departments of ophthalmology and otolaryngology at the University of Pittsburgh. The funds we provide from the Eye and Ear Foundation to support research are only made possible because of philanthropic support. So obviously we want to thank any and all regarding that, and certainly welcome anybody's interest in doing the same. Housekeeping, we do have chat disabled. So if you're trying to chat, don't get frustrated, we disabled it. But we do use the Q&A function, and we really want to hear your questions. So please put questions in. You can do them anytime. We'll save them to the end, and then I'll ask the questions to the panelists. And then please refrain from asking personal health questions. You can send those separately to Mr. Craig Smith, whose email is in your inbox. Please feel free to use this the show subtitles function for closed captioning, which we know a lot of people need. And then we'll be adding you to our email list for future webinars. You can always, of course, opt out of that if you like. So two of our favorite presenters, Dr. José-Alain Sahel and Dr. Leah Byrne, we're going to be talking about optogenetics, which is an innovative therapy that helps restore vision. And I'll let them share everything about that. But Dr. José-Alain Sahel trained at the Paris University Medical School and Louis Pasteur University in Strasbourg, and at Harvard University. He is an endowed distinguished professor and chairman of the Department of Ophthalmology at the University of Pittsburgh School of Medicine, UPMC. He founded in 2008 the Vision Institute at Sorbonne University and Inserm-CNRS in Paris, France, and the Vision Institute at UPMC, which opened in May of 2023, two large interdisciplinary research centers. His work has contributed to the understanding and preventing vision loss from photoreceptor cell degeneration. He conducted dozens of clinical trials in retinal conditions, including first-in-human trials of artificial retina, gene therapy, neuroprotection, and optogenetics, which you'll be hearing about soon. He published more than 750 peer-reviewed articles, holds more than 90 patents, and has co-founded a dozen startup companies. Dr. Sahel is the recipient of many prestigious awards, including the promotion to Officer of Ordre National de Légion, de honor the 2023 International Prize in Translational Neuroscience from the Max Planck Society, and the 2024 Wolf Prize in Medicine. He was elected as member of the Académie des Sciences Institut de France and fellow of the National Academy of Inventors in 2024. He was appointed the French Presidential Council for Science in 2023. It's the first I've read that much detail on Dr. Sahel, but I wanted everybody to appreciate again who we are honored to be hearing from today. And then Dr. Leah Byrne, who earned her bachelor's degree in neuroscience from Hamilton College and then studied as a Fulbright Fellow at the Karolinska Institute in Stockholm, Sweden. She earned a PhD in neuroscience from the University of California, Berkeley in 2007. And then Dr. Byrne worked with Ruth Kirstein, NRSA postdoctoral fellow for a Ford Foundation fellow at the University of Pennsylvania at UC Berkeley. Dr. Byrne started her research program at the University of Pittsburgh in 2017 and has been awarded a RPB Career Development Award, a Foundation Fighting Blindness Individual Investigator Award. And in 2023, she received the University of Pittsburgh Office of Innovation Emerging Innovators Award. Recently, Dr. Byrne was named a senior member of the National Academy of Inventors. Dr. Byrne is also the co-founder and CSO of Avista Therapeutics, a gene therapy biotechnology company based in Pittsburgh, Pennsylvania. So with all of that, we will first hear from Dr. Sahel, and thank you all again for joining us today on what I think you'll appreciate is one of our most interesting topics to discuss.
Well, thank you very much, Leonie, and thank you to the India Foundation to give us the opportunity, together with Leah, to share the status of this work. It's work in progress, trying to really promote function of photoreceptors in the retina and replace photoreceptors when they are no longer functional has been a challenge, and this is really relevant for multiple conditions, especially age-related macular degeneration, and for the sake of this today's presentation in retinitis pigmentosa. So I'm going to show some of the work we did both in Paris and Pittsburgh on optogenetics, explain what it is about, and then I will be very honored to hand over to Leah Byrne, who has really been instrumental in developing a novel approaches to optimize this approach and get it to patients more efficiently. So if we can move to the next slide, please. Just want to disclose that I have some financial interest in Gentile Biologics, who developed one of the trials I'm going to present, and another one in Inspiring Vision, which is developing an upcoming approach in optogenetics, which I'm not going to discuss today, but seems very promising. And I have a lot of activities, as Leonie was mentioning, filed many patents, and I'm also involved in many developments. Next slide. I want to acknowledge the collaboration of multiple people. So on the project I'm going to show, it's mostly collaborators in Europe, Denise Dalcara, Serge Picot at the Vision Institute in Paris, Boton Roska in Basel, with whom I've shared a couple of awards, and Isabelle Odo, who has been instrumental in the clinical trial. On the Pittsburgh side, we have benefited on the clinical side from leadership from Yossi Martel, who has been the principal investigator, and Michel Lalabek, who is identifying patients for these studies, and other collaborators that are on the slide. And as you will see, there is now an ongoing collaboration with Leah Byrne and Morgan DiLeo in Pittsburgh on developing the next generation of this therapy. Next slide, please. So as a reminder, the vision is relying on the activation of the retina, and specifically in the depth of the retina, what are called photoreceptors, cells that are able to capture the light and to trigger, after activation by light, electrical responses that are processed in the innermost parts of the retina, what we call the bipolar cells and the ganglion cells. And the ganglion cells are the cells that are connecting to the brain through the optic nerve. Then the processing of information is occurring at the level of the visual cortex, and this is where we see indeed. Importantly, the activity in the retina is not uniform. There is a more important activity in the central part of the retina, what we call the fovea of the macula, and there is a peripheral vision that is more fuzzy, and this is represented on the slide on the right, where you see the painting by Mrs. Henriot by Auguste Renoir, the famous Impressionist painter, where he represented the face of Mrs. Henriot with a crisp trait, quite crisp traits, where the rest of the body is fuzzy. This is what would happen if we didn't have eye movements to be able to direct the attention towards specific parts of what we are looking at. And only the central part of the retina, the fovea, has high acuity. So if we were not able to move our eyes, only the face of Mrs. Henriot would stay sharp, like it is on this painting, whereas in real life, we are moving constantly and we are able to get high resolution acuity wherever we want, but we don't notice that because we are not constantly thinking about where we are looking at. Next slide, please. There are multiple conditions that are affecting vision, and some of them are affecting central retina, and this would compromise the ability to see the face of Mrs. Henriot. And this is, for example, age-related macular degeneration, the most frequent cause of blindness across the world, but also Stargardt disease, for example. And then there is a group of conditions called retinitis pigmentosa, where initially dark adapted vision is impaired, and then peripheral vision, and eventually central vision, as you can see on the bottom part of the panel. Next slide. So if we try to describe the various therapeutic approaches that have been developed by many groups, including us over the years, the ideal situation would be to correct the gene defect. Let's say you have a gene defect that is affecting photoreceptors. Then you want to target this gene defect to replace the gene defect or to do what is called gene editing to correct the gene defect. And this is what a lot of work that Leah Byrne is conducting is really targeted towards correcting the gene defect, which is really the cure of a disease. Correcting the gene defect is going to prevent the cells from degenerating and from losing vision. Unfortunately, very often we cannot intervene early enough in the disease, or we don't know the gene defect, or there is not yet a gene therapy that is available to correct the gene defect. And then what we try to do is to avoid the progression of a disease, to protect the central vision. And this is what we do, for example, with neuroprotection. We have an ongoing trial based on work done by my team back in Paris, and actually Leah was part of that many years ago. And this is ongoing currently to try to protect the central vision. But then there is a stage of a disease where protection would not be enough because it's too advanced. And then you want to reactivate the part of the circuit that is still active. As I said, the ganglion cells, for example, are still connected to the brain. And we can do that using prosthetics, but we can do that also using optogenetics, which is going to be the focus of the talk from now on. Next slide, please. So when you position the various therapies, if you try to correct the gene defect, this would be gene therapy. This is what you have in the middle with a viral vector. You could try to replace the cells. It could be cell therapy, which is still in the making, but not very advanced. Or it could be electrical implants to really try to stimulate the retina. Optogenetics falls in the middle of that between viral vectors and electrical implants. Next slide. What is optogenetics? It is really using what you see on the top right side of a panel, proteins that exist in very rudimentary organisms like chlamydomonas, algae, whereby they have a protein that is able to respond to light. And when it is activated by light, it does open a channel or it does activate a pump at the membrane level of a cell. And this is inducing an electrical signal. And inducing an electrical signal in response to light is exactly what the retina is doing normally when we see. What you have on the left is what is happening at the level of photoreceptors. When light is absorbed by photoreceptors, proteins in the depth of photoreceptors, on the outer part of photoreceptors, are changing their shape. And this is activating a cascade of proteins. And these proteins, what we call a G protein coupled cascade, which is too complex to discuss now, is really inducing an electrical response to light, but importantly also an adaptation to multiple levels of lighting and an amplification of a signal. Classically, one photon, the smallest particle of energy that light contains, is able to activate through this amplification cascade 1 million channels at the cell membrane. So this is what is happening in our eyes constantly when we see. Of course, we don't notice that. It's an amazing mechanistic approach to vision that is enabling us to adapt to multiple levels of lighting and to react. In many conditions like retinitis pigmentosa, this machinery is no longer working. The photoreceptors either die or are no longer able to respond to light, and this is why patients are losing their vision. The idea of optogenetics is to use the proteins on the right to express them in the remaining cells in the retina to make the retina sensitive to light. As I said, you don't have the amplification that the normal retina is doing, but you have the ability to respond to light. This is this idea that several investigators, including us, myself and Boton Rosca, but others, have tried to deliver to the retina of animal models and then patients to restore vision. Next slide. So we tried different approaches. One was to reactivate the remaining photoreceptors, the cones. They have lost the ability to respond to light in advanced stages of the disease, but they are still connected to the rest of the retina, and potentially they could respond to light. So we did that, and actually we published that already 15 years ago in Science, and then we had several follow-up papers on that. And actually this is something which is still being explored currently by both Denise Dalcara in Paris and Boton Rosca, and we are bringing that into the clinic in the coming year. But this approach initially faced issues because of the inability we had initially, this is no longer the case, to target the cones efficiently. So we explored, and others did explore other cell types, bipolar cells, for example. The first to do that was Boton Rosca in 2008, and it showed that you could activate one visual pathway doing that. And we are currently working on that again with Leah, and hopefully we'll bring that to the clinic at some point. There are other cell types I'm not going to discuss, like the amacrine cells, but we decided to focus, for reasons I'm going to explain in in a moment, on ganglion cells in order to activate the cells that are directly connected to the brain in patients at advanced stages of the disease. And as you will see in the next slide, even if you look at different stages of a disease, you find that you have remaining cells in the retina. So when we wanted to work on the cones, we found that actually if you express proteins that are active and responding to light in the remaining cones in the retina, and you see that on the right panel of the slide using a specific protein, we are able to activate. And we showed that in patients with advanced stages of retinitis pigmentosa, we still have remaining cones. But as I said, we couldn't immediately translate that into the clinic. That's why we moved to ganglion cells. Next slide. And the ability to reanimate the cones seems promising, and this is why we are still pursuing that currently, as I described, but it would be relevant only to 20% of patients at best, although Botan-Roska believes that it could be more than that. Next slide. And so if the cones are lost, there are different ways to cope with that. One is, next slide, is to try to activate the ganglion cells. And this is what we have done, and what I will show in a moment. Next slide. Even in advanced stages of retinitis pigmentosa, if you look at the left of the slide, you see imaging we do in the clinic constantly when we see patients with multiple retina conditions. It's called optical coherence tomography. It's an optical imaging technology, and you are able to visualize the different cell types in the retina. So in blue, on the top, you would see photoreceptors. And in orange, on the top at the uppermost of the retina, this is the ganglion cells. And as you can see, these are different stages of the disease, coming from early stage of a disease to very late stages of a disease. Even at very late stages of a disease, the ganglion cells are still there. So the idea is to reactivate the ganglion cells in patients, even at advanced stages of a disease, to be able to respond to light. This was first attempted actually by a scientist in Detroit, Dr. Pan, in 2006. And next slide. He was able to demonstrate, this is on the left part of the panel, that if you express these proteins, and this protein initially had been identified by scientists in Germany, Ernst Bamberg, Ernst Nagel, and Peter Hegemann, and these proteins, which are the canonical protein that were identified initially in algae, he was able to express that in ganglion cells in mice, and he was able to observe visual responses. Unfortunately, the amount of light you need to activate this protein is enormous. It would be like staring at the sun in the middle of the day in the desert, and this is very unlikely to be practical, and most importantly, this is quite unlikely that it would be safe. So that's why we tried to use protein that would be more sensitive to light. So Ernst Bamberg, one of the inventors of this technology, developed a new protein that we tested in Paris with Denis Dalcara, and we were able to reduce the amount of light you need to activate, and we were able to show that this does induce visual responses in the brain in the animal. But still the amount of light was very high, so we decided to try to find a way to use an amount of light that would be safe. Next slide. And to do that, we collaborated with a team that was trying to define a way to reduce the amount of light. If the canonical optogenetic protein and the one I showed in the previous slide are responding in a wavelength that is in the blue or in the extreme blue of the spectrum, which does need a lot of energy and does induce a lot of energical activation of a retina. So the idea was to shift that into the red. Next slide. And to do that, there were two approaches. One that was developed by Roger Chen in San Diego, who got the Nobel Prize for protein engineering, the fluorescent protein. And this protein is shifted into the red. It's called ReachR. And another group at the MIT, led by Ed Boyden, developed a new protein called Crimson R. And Ed Boyden contacted me and Botan, and he offered to share this protein with us because he knew we were looking into a protein that would be shifted into the red. So this is the protein we selected and we tested. Next slide. So together with Denise Dalcara, we tried different constructions. She developed actually multiple constructions, including one using a vector that was actually developed in Berkeley by Leah Byrne, Denise Dalcara, John Flannery, and David Schaefer, the 7M8 variant. And we showed actually that there was an ideal construct that would be activating. This is what is in pink in the bottom left, the cells optimally. This is the one we tested in the clinic. Next slide, please. And we showed actually stimulating the retina of animals that were treated with this approach, but you could actually replicate different shapes and project different shapes on the retina, and you could calculate the theoretical visual acuity that the animal and potentially a patient could have. And what you see at the bottom is a mathematical model we developed out of that, and we could predict actually that there is a signature for each shape projecting on the retina, and this could be corresponding to a level of visual acuity, but in theory could be above a level of what we call legal blindness. This is work that was conducted by Grégory Gauvin, Serge Picot, and Ulysse Ferrari in Paris. Next slide. Then, based on a lot of studies we did in animal models, we were able to move into a clinical trial with GenTide therapeutics. The clinical trial was using 3 different components. One is a viral vector, a gene therapy vector, and you'll hear more about vectors by Leah Byrne in a moment, where we were putting in the vector the code for the optogenetic protein, the crimson R protein, plus a targeting sequence. This way, we wanted to really express the protein into ganglion cells, and we were able to make these ganglion cells sensitive to red light and to be able to respond to red light. Then we developed a stimulating system, goggles, that would send the light exactly onto this wavelength, would adapt to multiple levels of lighting, and trigger electrical response. And the third component of the study was a rehabilitation to train the patients to use this information. Next slide. So this was what was done on that study, injecting the vector into the eye, targeting the ganglion cells, using goggles to stimulate that. And we were fortunate to observe in the first patient in the study who was treated in Paris, that this patient who had been totally blind for the past 18 years was able to see object, to locate object, to count object. And then using a multiple electroencephalography, we were able to record from the brain of this patient and show that this patient was able to decode the information at the level of the occipital cortex, which is where vision is processed in the brain. So this was the first time ever that optogenetics was successfully applied to humans, not only in the eye, but in general. Next slide. Then we were able to replicate that in other patients. So this is another patient who was treated. She also was totally blind, and she was asked to count objects on the table. And as an example, the patient before was able to see the crosswalks also in the street besides counting objects. So here she's able to count how many pens are on the table. Now she's seeing the edge of the table. And then she's going to be asked to pick one object on the table. And as you can see, she does that precisely and quite fast. And so we are now putting together the data on the cohort of patients. We can move to the next slide. And part of the study is really correlating what the patients are telling us with cortical responses, recording from the brain. And as you can see, maybe I'm not going to comment on the slide, but at the bottom right, what you see in yellow is that only the part of the brain that is corresponding to vision, the occipital cortex, is activated during the experiment. Next slide. Then with Marlene Berman, who is in our team in Pittsburgh, who is a world leader in cognitive neuroscience, she developed a better way to record the activity in the brain. So there are now 128 electrodes that are outside. It's totally non-invasive to record the activity. Next slide. So if she's able to decode the activity corresponding at different shapes, and now we are putting together all this data, the ability to recognize shape and to have a signature of the brain level corresponding to that. So this is a first step. Next slide. We are not saying that this is the end of the road. It's just wonderful to be able to provide some level of vision to blind patients, but we can do much more. First of all, in parallel, I've been working on retinal prosthesis with Serge Picot and Daniel Palanker at Stanford, and now we have promising results with retinal prosthesis I'm not going to show today. We are trying to optimize the protein to limit the need for the goggles, or at least limit the amount of light that we need to stimulate that. We are trying to optimize the signal. We just had a few minutes ago a discussion with my former team in Paris with Valentina Emiliani, Emiliano Ronzetti, to really see how we can stimulate better. And interestingly, many of us have got into the field. John Flannery, who worked with Leah Byrne in Berkeley, had created 2 companies on that. Unfortunately, these companies have disappeared. There was another company that was formed in Switzerland that I think is also no longer active. Sheila Nierenberg in New York is working on an approach that is using optogenetics on ganglion cells. Ray Therapeutics is developing a new protein, and actually we are about to start a clinical trial in Pittsburgh using this protein to see how it works in a patient with end-stage retinitis pigmentosa. Next slide. And this is the landscape currently. Actually, we put that together with Yossi Martel some time ago. So some of the companies or groups are working on that don't exist anymore. The Retrosense Allergan, I think, doesn't exist anymore. The Novartis, Vedere, and Arctos don't exist anymore, but GenSight is still working on that. Bionic Sight is the company with Sheila Nierenberg, I think, and she's developing that approach, and she claims that she has very promising results. Nanoscope have presented data at multiple meetings. They have not yet published their data, but they claim to have promising results. And Ray Therapeutics is entering the clinic, actually has entered the clinic and is continuing that. So very promising landscape. The idea is really to move from just seeing objects and having shape recognition into even better. And we are also working on that, as we will describe in a short moment. Next slide. Yeah, and this is where I'm going to hand over to Leah. We are working on optimizing the approach and using better vectors, better delivery, and better stimulation pattern together with the team in Paris. And I think I'm going to stop here and to hand over to Leah to present the work she's doing currently on optimizing the vectors and making this approach even more efficient.
Thanks so much, Jose, for that talk. I always love to hear Jose present on optogenetics. And as Jose mentioned, I'm going to dial in a little bit now and talk to you about some of the specifics about, one aspect about optogenetics, sort of one thing that has to work correctly in order for optogenetics to work for patients. And that is development of the viral vectors that we're using to deliver these therapeutic genes to the retina. Just quickly before I get started, I want to acknowledge that I'm a founder and the CSO of a company, a Pittsburgh company called Avista Therapeutics. Jose is also a scientific co-founder of this company, and I'm a patent holder on some of the AVs that I'm going to talk to you about here today, so you can keep all that in mind. So as I briefly mentioned, for any type of gene therapy to work and for optogenetics to work in particular, we need these genes, these therapeutic genes that we're using to get to the correct cell type in the retina. So efficient delivery of therapeutic genes is really critical to the success of gene therapy. That's true for optogenetics, but for every flavor of gene therapy that we are investigating, including gene replacement or genome editing technologies as well. So delivery, yeah, it just really remains like a bottleneck in the development of optogenetics. So moving forward, we really need to work on improving delivery in order to improve therapeutic efficacy, to allow for cell type specificity. We heard Jose mention how optogenetic therapies have been tailored to different cell types, including retinal ganglion cells and bipolar cells and amacrine cells. So we need to develop better tools in order to specifically target those cell types. And then importantly, the vectors that we're working on, they can sometimes have off-target effects, like many drugs do. But in the case of gene therapy, it's really important that we're developing methods for gene delivery that are very specific to the cell types we want to target and are reducing, and we're working towards reducing off-target effects. And also, I'm going to talk a little bit more about vectors, but we're using viral vectors here to deliver gene therapies, and they didn't really evolve in nature to be gene therapy vectors. They evolved to be themselves, just to be viruses. And so they're not, they can sometimes be a little bit inefficient, which means that we have to increase the dosage that we put into a patient in order to get the effects that we're looking for. And that can sometimes cause problems like immune responses and toxicity. And, we don't want to have those side effects. And so we're working to create better vectors so that we can lower the dosage and reduce that inflammation and toxicity that we sometimes see. And just to drive home this point, this is a gene therapy a clinical trial that was run for a muscular dystrophy. And this is outside of the eye, so the issues are a little bit different there. But in some extreme cases, there can be some pretty bad effects of really high dosages of AAVs, including in this study where one of the patients actually passed away after receiving a really high dosage of AAV that was delivered systemically to the whole body. Of course, for retinal gene therapy, we're lucky that we don't have those same types of issues because the gene therapy is really localized to the eye. And the dosage that was used in this particular instance was really high, 3 × 10⁻14 particles per kilogram. So that's a lot of virus. So I've been talking about viruses. Viruses are the most typical vector that we use to deliver genes into retinal tissue. And there's one virus in particular that is most widely used for retinal gene therapies, and it's called adeno-associated virus, or AAV. And this is a picture of an AAV right here. We really like AAV for a few different reasons. One is that it is what's called replication incompetent, meaning that it can't make copies of itself. So it's not going to replicate, like it can't spread from person to person. Like when you get a cold, it's caused by a virus, it can spread from person to person. These AAVs are replication incompetent because we've removed all of the genome of the virus that it needs to replicate itself. AAV is non-pathogenic, so it doesn't cause disease in people. In fact, I think Lonnie said there's maybe about 100 or so people online today, and the majority of you have already encountered AAV in your lives at some point, and you just never even knew it. And in addition, AAV it can infect retinal neurons really efficiently. That's called tropism. And so that's really important that this AAV has the ability to transduce or infect the cells we want it to. And lastly, it can lead to permanent expression. So with a single dosage, we can lead to gene expression for the lifetime of a patient. And that's really the dream and the goal of gene therapy, is to cure disease with a single treatment. As I mentioned before, these AAV vectors, they have a lot of great properties, but they didn't evolve in nature for the purposes that we're using them for. And so they can sometimes be a little bit inefficient. They're inefficient for a couple of different reasons. One is that in the tissue that we're trying to target for optogenetics, the retinal tissue, there are structural barriers that prevent the AAV from moving through the tissue and transducing or infecting the cells we want it to. And that includes a layer that's on the surface of the retina called the inner limiting membrane. And so it acts as a barrier and prevents AAV from getting into the tissue. And then also inflammation is a big issue in retinal gene therapy. So because vectors can be kind of inefficient, we have to put in high dosages in order to get the therapeutic effect that we want. And when you put in a high dosage, you can trigger inflammation. And that inflammation can actually prevent the gene therapy from working. So antibodies can bind to the AAV and prevent them from transducing cells. But what's really cool is that we found out that over the past couple of decades, we have found that we can actually engineer new types of AAV vectors to enhance their performance. So we can take the vectors that nature has provided us and engineer them to be retinal gene therapy specific vectors. And so the way that we do that is by mutating the genes that encode for the protein shell of the capsid. So this purple box up here is representing a cap gene, which is the blueprints or the instructions for making this purple capsid shell. And so what we can do is introduce mutations into that cap gene, and that results in structural changes to the capsid. And those structural changes to the capsid then change the way that the virus interacts with receptors or barriers in the retina. And so we can mutate it in specific ways to improve the performance of the AAV and make it a retinal gene therapy specific vector. So in the past, we've done this successfully, and here's a couple of fun examples of how we've engineered AAV vectors to make them better for retinal gene therapy. So in one case, this is work out of my postdoctoral advisor, John Flannery's lab at UC Berkeley. We took this particular flavor of AAV, a serotype of AAV called AAV6, and we put 4 point mutations onto the surface of the capsid. And just by doing those small changes, we were able to change the way that the virus infects the retina. We changed it from not really infecting the retina at all to really efficiently infecting a particular type of cell called a Mueller glia. And then as another example, we took an AV2 serotype of AV, and we put a 10 amino acid insertion. So this is just a little peptide insertion on the surface of the capsid. And by doing that, we were able to improve the tropism of the vector and give it better ability to penetrate through structural barriers in the retina to transduce photoreceptors, which are the light-sensitive cells in the back of the retina. So the takeaway here is just that, we have several examples of how we can mutate the capsid and make it perform better for our particular uses. Now, one of the problems in the field has been, and this is a problem in many fields, a lot of the research is done in rodents. And sometimes what is done in rodents doesn't necessarily translate well into patients in the clinic. So in the case of these AAV vectors, again, we're actually injecting the AAV into the eye, as you see here on the left side of the screen. And on the right-hand side of the screen, you're seeing these fundus images or pictures of the back of the eye expressing a green fluorescent protein, this GFP. And the GFP is a marker, and it shows you which cells have been transduced by the vector. So if it's glowing green, it's a cell that was infected by the virus. And what you see in the mouse eye is with this virus that we made. These are all images with 7M8, that last virus I told you about. The mouse eye is transduced very efficiently. So you see that the whole field of the retina has turned green after injection into the eye. But in contrast, when you inject this virus into the eye of a non-human primate, which has an eye very similar to a human eye, what you see is that the pattern of expression is very different. So in this case, the pattern of expression is much more limited, and what you're seeing is a ring around the fovea, the high acuity part of the retina there. And so, yeah, it's not working quite as well in primates as it was in mice. Now, why is that? It's because the anatomy of mouse eyes is very different than the anatomy of large animal eyes and human eyes. So first of all, the mouse eye, excuse me, the mouse eye is just much smaller. So it's got a much smaller volume. And in contrast, large animal eyes like dogs and monkeys and humans, they just have much larger eyes. In addition, mice do not have a fovea, whereas humans do. The fovea is it's a pit that actually forms in the center of the retina, which is used for high acuity vision. And then in addition, I mentioned this barrier on the surface of the retina called the inner limiting membrane, or the ILM. And in mice, that barrier is actually very thin compared to large animals and in humans. So in humans, the ILM is a much more significant barrier, and it takes the virus a lot more to travel over that barrier, to cross that barrier. And then the immune response is much more significant in large animals than it is in mice. So mice are much more tolerant of, for example, some contamination or something like that, whereas humans are much more sensitive, and they would be more likely to produce antibodies and immune response against the viral vector. So in order to create vectors that are really going to work in patients and provide clinical benefit, provide benefit to patients, we developed a new pipeline for AAV engineering, AAV development, that we call Scavenger. I'm going to tell you a little bit about that right now. So Scavenger stands for single cell AAV engineering. And just want to point out that a lot of this work was led by a fantastic former postdoc of mine, Essine Ozturk, and Molly Johnson, who's now our Senior Director of Data Sciences at Avista Therapeutics. So I'm just going to walk you through here how the Scavenger pipeline works and how we're using it to develop AAV vectors that are going to work for patients in the clinic and really enable very efficient optogenetic gene therapies. So what we do is we create what are called libraries of mutated AAVs. And in these libraries, we have millions and millions of differently mutated AAV capsids. So each capsid has a slightly different structure. And each of the viruses carries a genetic barcode that allows us to track the performance of each one of the library members. All the viruses get pooled together and injected into the eyes of non-human primates. It's very important that we're doing this process directly in primates so that the end product is something that's going to work for patients. So after the pooled virus is injected into the eyes, each one of the viruses, it has a chance to transduce the retina. Some of those viruses, because of the mutations, are just non-functional, like number 1, that pink virus there. Some of the viruses might partially get into a cell, but they're not entirely successful, like number 2. But a minority, a very small number of the viruses are going to be able to get into a cell and traffic to the nucleus and actually produce a therapy like an optogenetic therapy. And those are the viruses that we are interested in finding out more about and isolating and then using for future studies. So after all the viruses have transduced the cell, then we take retinal tissue and we dissociate it, meaning we make it into a single cell suspension where we can look at each of the cells in the retina individually. And then we use this process called single cell RNA-seq to do 2 things. First, we identify what cell type it is. So it could be a ganglion cell or a bipolar cell or a photoreceptor. And we identify it based on its what's called a transcriptome profile. So we can tell what kinds of things the cell makes and then identify its cell type based on that profile. And then at the same time that we're doing that, we can quantify the performance of each one of the vectors in the pool by quantifying those that we use to tag each one of the viruses in the pool. So this really gives us really high quality data, at really high resolution, single cell resolution for millions of different viruses across millions of different cells. So this is really high resolution and high throughput data. Again, this is at single cell resolution, so we can actually tell the difference between viruses that infected one cell type versus another cell type. It's parallel and it's highly quantitative, and so it allows us to do these types of experiments directly in the same animals. So you're not trying to compare apples to oranges or compare what worked in one animal to what worked in another animal. Everything is done in the same animal, and that cuts down on variability a lot. And then we're doing just one screen for all cell types in parallel. So ideally, we'll be able to find viruses that are good for ganglion cells, viruses that are good for bipolar cells, viruses that are good for photoreceptors, all in the same experiment. And again, just want to circle back to this because it's really important. This pipeline can be run directly in a non-human primate model, which allows us to create viruses that are going to work for patients. And yeah, moving sort of the next thing that we want to accomplish with this is to look at cell type specificity, which is not just if it gets into a cell type, but can we find viruses that infect one cell type, but not the other cell types? And we want to do that, again, to cut down on off-target effects and really tailor a therapy to a specific cell type. So the first time we used a scavenger screen, we actually used it just to quantify the performance of existing viruses. This is just a proof of concept for the workflow. So we took 17 viruses and pooled them together, gave them each a barcode, used those to transduce a primate retina, and then looked at the results. And so these are UMAP plots. I love these plots because they show you all the data in one glance. So every dot in the plot there on the top represents a different cell that we analyzed in our workflow, and all the different colors tell you what cell type it is, and the black dots are cells that were transduced by an AAV. The bottom plot is just showing you the marker gene analysis. That's how we identified what cell type it was. So we use that data to create these heat maps that show you how well the different viruses in the pool perform across different cell types. So along the bottom part of that heat map, it shows you the different types of viruses, the different serotypes. And then on the y-axis, along the side there, it shows you different cell types. And the warmer colors mean that the virus is performing better. Cooler colors mean the virus is performing not as well. So I think you can by eye kind of pick out here that there was a subset of viruses that were working really well in these monkey retinas. And that's the type of data that we're looking for. So I said that we did the first screen with just 17 viruses. And once we saw that that worked, now we have started using this pipeline to actually create or engineer brand new viruses that the world has never seen before. And this version of the pipeline we call scavenger hunt, which we worked really hard on finding that fun name. So it stands for high unbiased throughput scavenger screening. And yeah, we're using this to screen really high throughput libraries. So just as an example, this shows you the workflow, which is very similar to the workflow I showed you before. We're pooling libraries of viruses, injecting them into non-human primates, and then analyzing millions of cells in parallel. And then we rank the performance of those millions of viruses at the single cell level. And this is some of the output of one of these studies. And it's the same type of plot I showed you before, where every black dot is a cell that was transduced by a virus. Now here's a heat map that resulted from one of our first scavenger hunt screens in a non-human primate. And so what you're looking at here is the results from thousands of different viruses that we identified that have the potential to transduce primate retina. And then if you focus in here on, these have all been ranked according to their performance. And so at the bottom right-hand corner here, we're just zooming in on the most promising variants that came from the screen. So these are the ones that are the most potent, that had the best ability to transduce across all the different retinal cell types and across all the regions of the retina. And then so once we identified from those thousands of variants, a couple hundred that we thought we really wanted to take a closer look at, we just we ran another scavenger screen where we had just a couple hundred variants in the pool, and then we quantified their performance, and that's what this heat map is showing you. So it's showing you how hundreds of different variants, which are the different columns in this heat map, are performing across different retinal and other cell types, which are shown here along the side. And if the data looks a little confusing, you can just look at the top row there, which shows you the performance across all the cells combined. So it's the sum of the data below it. When we compare the performance of our new variants to wild type AAVs, we can see that, first of all, the wild type AAVs, so this is AAV9, one of the flavors that exist in nature, and then AAV2, a serotype that also exists in nature. Those are not performing very well, so they're at the left side here of this heat map. In comparison, 7M8, which is one of the variants that we've engineered previously, it definitely outperforms the wild type serotypes as we expect it to. K912 is another variant that we created previously. It also performs better than the natural serotypes. But we have also created this new tier of AAV vectors that outperforms the previously generated ones by quite a lot. One of our top vectors is one that we call ATX002, and this is one that is being developed in the context of Visa Therapeutics, that startup company that we mentioned earlier. And this top performing variant we have quantified to be about 30 times better than 7M8. So it's a significant improvement in the performance of this AAV vector as a result of the engineering process we went through. And we think that this can translate into more efficient optogenetic therapies or other types of gene therapies for patients, and will allow us to reduce the dosage and reduce those off-target effects, and reduce inflammation, and hopefully really lead to better outcomes for patients at the end. So these are images showing you how that one vector that we've identified as a top performing vector called ATX002 is performing by itself in a non-human primate. And so what these pictures are showing you is GFP expression, again, this expression of this green fluorescent protein marker gene after we've injected the variant into non-human primate eyes. And so we're comparing 7M8 and ATX002 here. And what I hope you can appreciate is that the ATX002 leads to brighter expression and wider expression across the expanse of the retina compared to 7M8. And what's kind of cool is that ATX002 also performs well outside of the non-human primate eye. So if you take it and you use this vector back in a rodent model, like a mouse, it actually still outperforms 7-8, which is very interesting. And then we also wanted to check and make sure that the virus was going to work in human cells, of course. Wanted to make sure that we hadn't, mysteriously created a vector that was really specialized for non-human primates. So we also tested it in the context of a human retinal organoid. So these are cells that have been collected from patients and grown into these retinal-like tissues in a dish. And so we can see that the ATX002 does transduce efficiently across many types of tissues, including in human cells. So really quickly, I'm just going to run through here. We also used ATX002 to deliver a therapeutic gene called RS1. This would be used specifically for a gene replacement for a disease called X-linked retinoschisis. And what we did here was compare ATX002 to AV8, which has been used in previous clinical trials for this particular disease. And the takeaway here is that ATX002 is expressing about between 6 and 300 times more of this therapeutic retinoschisin gene compared to AV8. So we're getting a big bump in gene expression, which is good there. And we can see that it infects a lot of different cell types, and that for the outcome of expression with this vector is that we're getting retinoschisin levels that are pretty close to what a patient, what would be normal in a human retina. So here's a picture of retinoschisin expression in a retina from AV8 versus ATX002. AV8's on the left side and ATX002 is on the right. And I hope that you can appreciate here that there's more of this retinoschisin, which has been labeled with a fluorescent marker in the ATX002 injected eye here. Again, takeaway here is that with these new engineered vectors, we're getting greater expression of our therapeutic proteins, which we hope translates into better outcomes for patients. I think I have maybe 1 more minute left, so I'm just going to really briefly plug a new effort that we've been working on here in the Byrne lab in collaboration with the Betagon lab, and just want to highlight Sushma Sapa, who's the scientist leading this. We're trying really hard to share the data from these studies and to make these tools available to other folks. So we've created something called the Pittsburgh AAV Atlas, and it's a public database for AV vectors, and we're hoping to launch this extremely soon. Please check our website if you want to have some fun looking at vectors and how they perform. But online, we're providing information for how all these vectors how well they're transducing in primate cells. And we're providing a bunch of information, including how we made them and how we do the quantification and how the immune response looks in these animals. And it's all very interactive. So you can check out serotype by serotype how each of the viruses work. So if you are ever curious about AAVs and want to take a look at that database, please check out the Byrne Lab website. And I think I'll stop there because I think we've seen that we have some questions. So I'll hand it over to Lonnie.
Well, thank you both so much. Yes, we do have a lot of questions. I think I'll start off because I think I can summarize some of the questions that I'm seeing with just sort of one sort of question for you both. Long question. So this is amazing work. It's amazing work because, and so everybody clearly understands the gravity of this. We're looking at all of these photoreceptors that are in the back of our eye that degenerate because of these conditions, these inherited retinal degenerative conditions, as well as even our age-related degenerative conditions like macular degeneration, other things which so many people have. And all of those photoreceptors can't be replaced. No way to do it right now. So this technology is a way to use a different photoreceptor from protein, putting it into the nerve cells that work with those photoreceptors that should be there in our eye, providing light in a way that gives it a different image. So with all of that, what I'm seeing here is, and this is, I think, to some of the questions, what are the limitations, or are there, how much further can we take this in terms of the different conditions that can be treated, the level of acuity of our vision that we can receive? Do you have some ideas from what you've seen so far, or maybe what you understand about what's actually happening with the science you're applying, to what those limitations are, or are they at this point still limitless?
Well, I mean, it's a multiple question. So the work that Leah is doing, which is very technical, but at the same time very targeted, is going to enable a much better selectivity. You don't want to treat every cell in the retina. You want to really target the cells that are going to be the ones that give the highest resolution. You want accuracy. If you think about the pictures, pixels, you want the most accurate, the highest level of pixels in the imaging you get. And for that, you need accuracy. You don't want to stimulate things cells that don't need to be stimulated. So this is one part of it. Another part is to optimize the proteins. I mentioned a few that have been developed, and we are working on others, how to make them even more selective, also more sensitive to light, which would enable us to use more classical goggles than the ones we are using currently. So this is one avenue. Currently, the vision that is achieved is moving from blindness to being able to see objects, to grab objects, but it's not reading. So this is something we want to achieve through all these steps, and we didn't get into all the details of the program if it was already quite detailed, but really trying to really enable the retina to perform as close as possible to the normal vision. But we will never get to normal vision. It will be always lower vision than normal, but hopefully useful vision that people can use to see faces, to recognize emotions. Currently, it's only targeting conditions where the optic nerve is functional. So people that are affected with optic nerve atrophy or advanced glaucoma or trauma, this cannot be done this way. There are other approaches we are working on, but it's beyond the scope of this webinar to target these conditions. So it's really targeted for disease of the photoreceptors, potentially inner retina, but as long as you have ganglion cells connected to the brain.
Thank you. That does answer quite a bit of it. And Dr. Byrne, I think you are adding to the opportunity by the fact that you're creating so many different viruses that can get into all these different places. So right now, are the limiting factors those types of proteins that can be used, as well as the where you need to get them with those with the viruses that we have available today?
I would say that there's opportunities on all fronts. Yes, we've definitely seen a lot of potential for gene therapy, but I think there's ways that we can improve on multiple levels. So we can definitely improve gene delivery, as I was talking about in my talk today. José mentioned that the proteins that are being delivered can also be improved. There's many different ways, though, that I think we can work on gene therapy to make it even more beneficial. The surgical approach, for example, is something that we're also working on. We talked about rehabilitation and retraining the brain to see, and even things like maybe multifactorial types of therapies where we could support cell survival on one hand and then replace a gene that's not functioning on the other hand. So I think we're really just scratching the surface of what gene therapy can accomplish. There's a lot of work to be done, but there's a lot of fantastic people working on it, and I think we're going to see a lot of development in the coming years.
Okay. And to summarize some of these questions too, of course, right now there's a clinical trial with this, but and the question is, how long it'll take to get out of a clinical trial into regular practice, and then affordability. I think those are two things that maybe to get your thoughts on for either of you.
Yeah, so the GenSight trial is finished. We are now working on the next generation of this therapy, but this is going to be another trial. The Ray Therapeutics trial is about to start in Pittsburgh, and we are currently looking into two approaches to reactivate cone photoreceptors. All of these are early stage trials. Once the trials are shown to be safe and efficient, then you move into another phase, which is a larger size of a trial. And this is only after this trial is completed that the agencies, the FDA, or the the EMA in Europe can approve a therapy to be available to patients. Our goal is to make it totally free for patients. I mean, the goal, trials are free anyway. Nobody's paying anything for the trial, only the sponsors. And the goal, like we have currently with Luxturna for gene therapy, is to work with the payers to make it totally free for the patient, because obviously this would be an amount of cost that would be beyond acceptable. And so it's absolutely our goal is to make it available for everyone.
And you maybe see Dr. Sahel, or somebody says they have known you for 35 years, but...
Oh, yes, Henry, we worked together when I was a fellow at Mass & in Boston, and I have wonderful memories of Henry. So happy to reconnect with him. I hope you can share my address with him. I would love to continue. Well, Henry was an amazing tech and student. I know he has had a wonderful career afterward, and his question, of course, we don't work only on Usher, we work on any type of retinal degenerations. Actually, the vectors that Leah is developing are targeting multiple conditions, as she was showing. And so we are still working on all of that. So not much change since 40 years ago, more work to do. Yeah.
Yeah, and I think that summarizes a lot of the other questions too, which, I know we're running short on time, but I think a lot of the other questions too were what types of diseases, Ushers and Stargardts and other. Essentially, I think, is it safe to say if it's something that degenerates those retinal cells, those photoreceptive cells, that this has potential to be an answer for those conditions? Is that safe to say?
Yeah.
Okay, I think that, so that kind of answers a lot of those questions. Now, we also have questions about things that are not related to the photoreceptors. Those are for maybe other talks, or maybe we can answer you offline, because we certainly are working on many things. I see that there's a lot of concern over the optic nerve, and we're doing a lot of work in that area, of course. So wonderful stuff, very exciting. Certainly a lot of information, but all incredibly worth hearing about and knowing about. And I know that we'll get a lot of views on our website. So, but thank you everybody for attending. Thank you, Dr. Sahel, Dr. Byrne, for the wonderful information. It is encouraging as can be, and we need to keep this type of research going. We appreciate everybody's efforts in doing that through philanthropy, but also, by letting people know how important this is to you in terms of what we're, what we support as many institutions associated with funding that we receive from various sources, how important it is. Thank you all, and we wish you all a good day.